Hello. Today we're going to discuss the relationship among different alcoholic beverage glasses using a phylogenetic tree. You can think of a phylogenetic tree like a family tree. It shows the relationship among species, objects, genes, etc. based on similar characteristics among the different objects. Here we can see the different objects we will be putting into our phylogenetic tree. Each one of these glasses is considered an OTU, or an operational taxonomic unit. These are the specific objects that are part of the phylogenetic tree and are typically arranged based on differences in their DNA sequence. In this example, I will be grouping the glasses based on similar characteristics versus DNA. Here's how I've chosen to build this phylogenetic tree. I'm going to break down some of the different related terms and explain why I grouped the glasses as so. First, this pink plastic bucket serves as the outgroup for our tree and allows our phylogenetic tree to be rooted. An outgroup is an OTU or object in the phylogenetic tree that is known to have diverged earlier than all of the other OTUs, so all of the other OTUs share this as a common ancestor. A rooted tree shows the distance between different OTUs or glasses, and the order of divergence can be determined by comparing the different branches to the outgroup. The branches in this phylogenetic tree are the horizontal lines and are indicative of the time it took for the different objects to evolve. More closely related items will have shorter branches, as seen between the red and white wine glasses. There are two different forms of evolution that can be seen in our phylogenetic tree. First, anagenesis is accumulated genetic changes over time within the same lineage. If we look at this branch, we can see where anagenesis is occurring. A 16-ounce beer glass that is here is slightly different from the one here. Cladogenesis is evolution that involves the splitting of one lineage into two. We can look at the internal nodes, which occur right before a split in the genetic line, to see where cladogenesis occurs. If we follow these branches to the terminal nodes that exist at the end of the branches, we can see that they are connected to the two new glasses. In this case, the 16-ounce and 22-ounce beer glass. Looking back at the internal node, we can see the most recent common ancestor between the 16-ounce and 22-ounce beer glasses here. As to why I grouped the glasses as so, let's take a look at this first split. I chose this split based on stemmed glasses versus stemless. The 16-ounce and 22-ounce beer glass both hold beer, lack handles, and are stemless, so I grouped them together here with the most recent common ancestor. Here, the two branches are split into a group of wine glasses and a group of mixed drink glasses. If we follow the branch, here, we can see another split into two different groups, stemmed and stemless wine glasses. You can see the branches between the red and white wine glasses are shorter than the branches between the stemless and insulated stemless wine glasses. This shows that the red and white wine glasses are more closely related than the stemless and insulated stemless wine glasses. I chose to have the stemless wine glasses branch off here because even though they lack stems, they have the exact same shape and function as the wine glasses. Therefore, I thought these two glasses were more similar to wine glasses versus beer glasses. Finally, looking at the branch that leads to the three mixed drink glasses, we can see daiquiri glasses branched off and then margarita and martini glasses split. All three of these glasses contain different drinks that are made with different liquors and ingredients. Therefore, I grouped martini and margarita glasses as being more closely related due to their similar glass shape and taller stem lengths than the daiquiri glass. Thanks for watching, and here's a list of my references.